Hi, welcome back to part 2 of chapter 8 for photosynthesis. Okay, time to take out your worksheet again for photosynthesis. During the light-dependent reaction, it involves two large complexes called photosystems. They are made up of light harvesting complex, which is where chlorophylls are bound and transfer energy from the sun to the electron carrier. So that's why you see light coming through and it goes through those green dots, okay? And there's electrons involved. So that's where the sunlight is coming through. Those green dots are your chlorophyll, and then that's going to cause electron transfer, and we'll talk about that in detail in a bit. The other main part is called the reaction center right here, where much of the photochemistry of turning light energy into chemical energy takes place in order for photosynthesis to happen. So go to number 2 in your worksheet and we can label the photosystem protein complex together. In A, it is referring to the thylakoid membrane right here. Thylakoid membrane, A. Just like how protein complex sits in the mitochondrial membrane for cellular respiration, the one for photosynthesis integrates into the thylakoid membrane. So within those circular stacks of thylakoid, they are actually has that phospholipid bilayer that we were talking about. Now for B, it is pointing to pigment molecules, these green dots all over here, and I would add chlorophyll as well. For C, it is pointing to the reaction center, which is this blue area in the center of the photosystem complex right here. For D, it is referring to the light harvesting complex, which is right here. Light harvesting complex, the green portion, the lighter green portion right here. And for E, it is the primary electron acceptor. This pink red oval right here is your primary electron acceptor. Okay, so remember the final electron acceptor for cellular respiration was oxygen. For photosynthesis is actually something within the reaction center that accepts that pair of electrons. For F, it is referring to sunlight or light. Okay, but I would put sunlight in there just so you know the energy is coming from the sun. For G, it is a chemical reaction that donates electrons to that special pair of chlorophyll pigments right there. Okay, the chemical reaction is what I want you to remember and write down. Water is split into oxygen and hydrogen. Okay, so you just write this reaction down for G. So you see how already that this is a reverse of cellular respiration. In cellular respiration, you have, remember the last step of electron transport chain was oxygen serving as your final electron acceptor to make water. Remember we had in cellular respiration this plus water to make water. Okay, it doesn't matter the numbers, but that was the reaction that happens at the last complex of cellular respiration, electron transport chain. Now for photosynthesis, we're going reverse. We're going to split water to get electrons back. Now we're going the other way around in that sense. Okay, so this is why plants are able to make oxygen for us, because when they split the water molecule, they make oxygen and hydrogen. Hydrogen is where we're using it to pump things that we'll talk about in a bit. And also that's where we're getting your electrons back. Oxygen is what's going to be released from plants as a byproduct. Even though oxygen is a byproduct for them, remember for us, it is an essential molecule for survival because we need to breathe in oxygen in order to do cellular respiration, or else no ATP can be made. All right, so that should be all the labeling for number 2 in your worksheet, okay? All right, and it'll be a bit before we get to number 3. So put that down for a little bit before let me finish this slide. So there are actually 2 of these photosystems for photosynthesis, and they are both essential parts of the plant's electron transport chain. One is called photosystem 1, and the other is called photosystem 2, or PS2 versus PS1. They do not occur in sequential order as the numbering goes, okay? Photosystem 2 actually has to happen, or something happens to photosystem 2 first before you get to photosystem 1. So we'll see, what happens for light-dependent reactions with these 2 different photosystems in a bit, and they differ in what they oxidize and what they reduce as well. Okay, and I will emphasize what they oxidize, what each of those reduce, because you will have to know that for the exam. All right, so like I said, they are both major components of the photosynthetic electron transport chain, and these are just the 2 major parts. There are, the other protein complexes within the electron transport chain that we don't have to identify, okay? All right, so let's go through the steps of your light-dependent reactions next. So let's pull out your worksheet again. Go to number 3 on your worksheet, item number A. Item letter A. The first step of light-dependent reactions happens in the photosystem 2. Remember, this is not photosystem 1 that comes first in photosynthesis. It is not in numerical sequential order. Photosystem 2 has to have something that happened to it first, okay? So that's why first it is the photosystem 2, or PS2, and this is where a photon, okay, so photon of light is absorbed by a pigment called chlorophyll and passes energy to other chlorophyll pigment throughout the photosystem until it reaches your reaction center. So those are the 3 blanks in your number 3, item A, okay? The first blank is photosystem 2, and then the other 3 blanks I have circled on the PowerPoint, okay? So for item B in your worksheet, then a special pair of chlorophyll pigment within the reaction center will undergo an oxidation process called photoact and converts light energy from the sun into an excited electron state. Okay? Now, for item C in your worksheet, the reaction center then delivers that electrons from the special pair of pigment to the primary electron acceptor, which follows through with the rest of the electron transport chain here to travel to PS1. Okay, PS1, that's where it travels. And for item D in your worksheet, the electrons in that special pair of pigment is replenished after each excitation reaction by splitting of a water molecule, which was that reaction I asked you to write down in number 2G. Okay, water, it's split to replenish that pair of electron that's going to be donated to the special pair of chlorophyll pigment. So let me summarize before we move on, okay? So here is that photosystem 2. The photon of light hits one of these chlorophyll pigments on photosystem 2. A bunch of excitation reaction happens within these chlorophyll to transfer the energy to this special pair of chlorophyll pigment. And once that chlorophyll pigment is excited, it can undergo oxidation reaction, okay, where the electron, where it will excite the electrons and be accepted by that primary electron acceptor. So all this green dot is doing is just passing that energy so that this special pair of chlorophyll pigment has the energy to excite that electron to get to the primary electron acceptor states. Without all this transferring of energy, that pair of electrons can never go to the primary electron acceptor. Okay, and once that electrons is gone because it's been passed on to the primary electron acceptor, this electron needs to be replenished, and it is replenished by this reaction right here where you have water splitting into oxygen and hydrogen and replenishing this pair of electrons again. Okay? All right. So our ultimate goal here is to allow the energy from the sun to travel in form of an excited electron that will be passed on to an electron carrier, NADPH, which will then travel to the light-independent reaction, which is a second part of photosynthesis called the Calvin cycle. So as the electrons are moving through the proteins of electron transport chain between photosystem 2 and photosystem 1, they will use energy to pump hydrogen ions from the stromal side of the thylakoid to the thylakoid lumen. Okay, again, we are going to create that same hydrogen ion gradient and have chemoosmosis going on to make ATP. Because remember, one of the other things that goes into your light-dependent reaction was ATP. So the plant has to have some way of making ATP as well. So that's where the hydrogen ions going to be pumped from the stromal side into the thylakoid lumen side, which will then go through, as you know it, your ATP synthase. So for item number E in your worksheet for number 3E, the hydrogen ion is pumped through the electron transport chain from the stroma into the thylakoid lumen to create a concentration gradient that will be used to power ATP synthase in production of ATP molecules. Remember that one of the products that will be delivered to light-independent pathway other than NADPH is ATP. So this is where ATP is made. Both ATP and NADPH has to be delivered to your light-independent pathway, which is your Calvin cycle, in order to finish up your photosynthesis. If these two things aren't delivered to your photosynthesis, then you're never going to get glucose in the end. Now, so for item F in your worksheets, next, another photon of light is absorbed by PS1. Okay, now this is PS1, not PS2, where energy is relayed to the reaction center and send electrons to reduce NADP plus to NADPH. Okay, so make sure you write NADP plus NADPH. Okay, so that's the major difference between this electron carrier and the one in cellular respiration, just that letter P. Okay, so the electron carrier for photosynthesis is NADPH. Okay, as you can see from your PowerPoint slide there. So here's the light-dependent reaction in a nutshell. For PS2, they are capturing energy from the sun first, then turn it into chemical energy in form of ATP by creating a hydrogen ion gradient, as you can see here. And I'll summarize it in another picture as well. The PS1 will then capture another photon of energy so that it can reduce NADP plus to NADPH in order to pass electrons to the next stage, which is your light-independent reaction. Both of these systems work together to form ATP and NADPH. So that's why you have two systems going on, because they are doing different things, okay? PS2 is making ATP, okay, with the hydrogen ion gradient. PS1, you're reducing NADP plus to NADPH. And water is only split in PS2. Okay, water splitting only happens within PS2, not PS1. So this is a good summary diagram that illustrates the process of light-dependent reaction again. Now you have this photosystem 2. A photon of light hits one of those chlorophylls within photosystem 2, okay, which then gives energy to excite this electron, which this electron was originally donated from a molecule of water. When a molecule of water splits into oxygen and its hydrogen molecules, you have electrons that can be donated to that special pair of chlorophyll pigment. And once the electron is donated to your primary electron acceptor, then it goes through the electron transport chain. You don't have to know the names of those different ingredients within electron transport chain. Through here, you're actually pumping hydrogen ions again, okay? And that's what's going to produce your ATP molecule from photosystem 2. Once that pair of electrons reach your photosystem 1 right here, so photosystem 1, okay, so that's why photosystem 1 does not need water, that does not need to split water again for donation of electron, because electron is coming from photosystem 2. Photosystem 2, this pair of electron is going to follow and drop to your photosystem 1, okay? It just need that photon of light as energy again to excite that pair of electrons up to be up here, and hence go through another round of electron transport chain down here to reduce NADP plus to NADPH. Okay, so two goals here. PS2 makes ATP, PS1 reduces NADP plus to make NADPH. Okay, so this is just the light-dependent reaction, which is first phase of photosynthesis. Okay. So this is sort of pictorial diagram for you to easily understand what is going on. And don't forget, this is your water splitting to replenish that special pair of chlorophyll from photosystem 2 with electrons. Okay, now we are ready to move on to the light-independent reaction. It's called light-independent reaction because it doesn't involve any photons of light. That's why the other phase was called light-dependent reaction, because you needed 2 photons of light, one for photosystem 2, one for photosystem 1. But for light-independent reactions, you don't need any photons of light to finish the rest of the process. Similar to Krebs cycle in cellular respiration, but kind of in reverse order, okay? Because now we're trying to make the intermediate that will make glucose. So remember, in Krebs cycle from cellular respiration is when we are moving carbon dioxide from our acetyl-CoA molecule and all those intermediates in between. You're removing carbon dioxide. That's why you have to breathe out carbon dioxide. For Calvin cycle, which is your light-independent reaction in photosynthesis, we are adding carbon dioxide to our intermediates. That's why I say it's kind of like a reverse process of Krebs cycle. Krebs cycle, you're removing carbon dioxide. For Calvin cycle, for photosynthesis, you're adding carbon dioxide to your intermediates. This is why plants need to breathe in carbon dioxide, okay, through their stomata, because they need it during Calvin cycle. The carbon dioxide will travel from their stomata and diffuse into the mesophyll cells, okay, and then travel to the stroma portion of the chloroplast within those cells. The Calvin cycle doesn't take place within thylakoids, it takes place within the stroma. Okay, so that's another difference. Okay, so you have light dependent reactions happening within the thylakoid, okay, which are those green circular stacks of pancake-looking things. For Calvin cycle, it happens within the stroma, okay? Stroma, not within the thylakoids anymore, okay? All right, so now go to number 4 in your worksheets. We're almost done here. We can fill in that light independent reactions are also called the Calvin cycle. Okay, Calvin cycle. Don't mistake this with Krebs cycle. It is divided, here's Calvin cycle in spelling in case you need it. It is divided into 3 substages. Okay, go to item A in your worksheet. The first stage is called fixation, where CO2 will react with RUBP. Okay, that's the reactant right there. And then using an enzyme called Rubisco. Okay, so this is your reactant, RUBP, and carbon dioxide. And using the enzyme called Rubisco, they can turn and make 3-PGA. Okay, 3-PGA. You don't need to know the entire name. Just know it as 3-PGA molecule. This is the first intermediate within the Calvin cycle. So if you see, this is the Calvin cycle right here. This is that 3-PGA intermediate that you're making by reacting CO2 with RUBP through Rubisco enzyme to make 3-PGA. Okay, which is now 3 molecules of carbon dioxide will produce 6 molecules of 3-PGA. So that's why you have 3 molecules of carbon dioxide right there, which makes 6 of these 3-PGA molecules. Now go to item B in your worksheet. The second stage is called reduction, where ATP and NADPH from light-dependent reactions are used to reduce 3-PGA into 6-glyceraldehyde 3-phosphate. Okay, so again, this is the second stage right here. Okay, second step is called reduction. Oh, excuse me, I crossed it out. Let me erase that. Okay. So it's reduction reaction, and you're turning the ATP and NADPH using those energy provided and electrons provided to turn your 3-PGA into glyceraldehyde 3-phosphate. Okay, so that's why you see ATP being inputted into the Calvin cycle, and then NADPH being input into the Calvin cycle, and NADPH then gets oxidized back into NADP+. And this then goes back into the thylakoid to pick up more electrons via your PS1, okay? So now you're making this 6 molecules of glyceraldehyde 3-phosphate. All right. And you're making 6 of those glyceraldehyde 3-phosphate, okay? Go to item C in your worksheet. The third step is called regeneration, where one of those glyceraldehyde 3-phosphate goes into the cytoplasm of the plant cell to form other compounds needed by the plant. For example, glucose molecule. The rest of the 5 glyceraldehyde 3-phosphate actually goes and remain in the Calvin cycle to regenerate more RUBP, so that the cycle can go on and on and on, because not only are you making your products, you're also regenerating those reactant via your Calvin cycle, so you can keep picking up more carbon dioxide molecule to make 3-PGA. 3-PGA then turns into glyceraldehyde 3-phosphate, which then makes more RUBP and start the whole cycle again. Okay? So notice that with 3 molecules of carbon dioxide, we can only produce 1 glyceraldehyde 3-phosphate. Okay? 3 molecules of CO2 makes only 1 molecule of glyceraldehyde 3-phosphate. So that's why 3 turns of Calvin cycle is necessary to make that 1 glyceraldehyde 3-phosphate right there, which is only half of a glucose molecule. Remember, in cellular respiration, during the first few steps of glycolysis was when we split the glucose molecule into 2 parts. So now we are combining the 2 parts back together to form glucose. So with 3 molecules of CO2, we go through Calvin cycle 3 times to produce 1 molecule of glyceraldehyde 3-phosphate. Since 2 glyceraldehyde 3-phosphate is necessary to make 1 glucose molecule, you need a total of 6 turns of Calvin cycle in order to produce 1 glucose molecule for photosynthesis. So this should be the answer that goes to item letter D in your worksheet, okay? 2 glyceraldehyde 3-phosphate is required to make 1 glucose, while we need 3 turns of the Calvin cycle to make 1 glyceraldehyde 3-phosphate. That means we need 6 turns of the Calvin cycle to make 1 glucose molecule, okay? So here's a good summary image for the entire process of photosynthesis. Okay, you have your thylakoid right here, represented. Remember, the chloroplast looks like there's stacks of these membranes. So we're zooming in into one of these stacks right there. And within the thylakoid membrane, you have your photosystem 2, PS2. 1 photon of light comes through, which excites that pair of electrons to your primary electron acceptor. We can replenish that electrons through splitting of water molecule, which then produces oxygen as a byproduct. This electron then travels down the electron transport chain to photosystem 1. In the process of traveling through this electron transport chain, that you don't have to know those blue integral protein names, okay? Their job is to pump hydrogen ions from the stroma, okay, to the thylakoid lumen, okay? So the stroma has a lot of hydrogen ions around, and it's being pumped into the thylakoid lumen. And all this buildup of hydrogen ions, just like cellular respiration, is going to go through your ATP synthase to make molecules of ATP. ATP will then travel to Calvin cycle. Okay, so when this hydrogen ions goes through, all these hydrogen ions goes through ATP synthase. These hydrogen ions is what's going to be replenished back to this side. So you can keep doing this pumping of hydrogen ions into thylakoid membrane. Then hydrogen ions goes out the ATP synthase to make ATP. All these hydrogen ions recycles back here. Again, do the process all over again. So that's photosystem 2. Goal is to make ATP molecule. Now photosystem 1, you have this pair of electrons from photosystem 2. Another photon of light has to come through and excite the chlorophyll, which then excite this pair of electrons that has just arrived from photosystem 2, which excites it into your primary electron acceptor, okay? And this pair of electrons from the primary electron acceptor will then travel through 2 short electron transport chain proteins to reduce your NADP plus to NADPH. So that's the whole goal of that electron from photosystem 1. NADPH then travels to the Calvin cycle here so that they can deliver electrons through to intermediates within the Calvin cycle. So now we are at the light independent reaction, which is the Calvin cycle. Remember carbon dioxide, this is where carbon dioxide is inputted, okay? 3 molecules of it is inputted into the Calvin cycle, reacts with RUBP through the enzyme called Rubisco, okay? And it makes 3 PGA, and then 3 PGA gets to be made into glyceraldehyde 3-phosphate using your ATP and NADPH. 3 molecules of CO2 makes 6 molecules of this glyceraldehyde 3-phosphate. 1 comes out to make your half glucose. The other 5 gets recycled back into RUBP to continue going through this Calvin cycle again, again, and again. Okay, so it needs to go 3 more times to make 2 G3P or glyceraldehyde 3 phosphate. So 6 Calvin cycle gives you 2 G3P, glyceraldehyde 3 phosphate, to give you 1 molecule of glucose. Okay? All right, so that wraps up the light-dependent reaction. The light-dependent reaction, I'm waiting for the laser pointer to come up. It's not coming up. I don't know why there. Light-dependent reaction, and then you have your light-independent reaction that will ultimately make your 1 molecule of glucose via photosynthesis. I will upload this animation video to eCampus for you to see these reactions visually, which will help you understand it even more. Okay? All right, so this illustrates here that how photoautotrophs and heterotrophs have to depend on each other to survive. Okay, it's a cycle process. While photoautotrophs uses photosynthesis to build glucose, or carbohydrates for us to eat, they need CO2 in order for photosynthesis to happen. So they need CO2 from us to complete the process. On the other hand, we heterotrophs are dependent on consuming and metabolizing those glucose molecules from photoautotrophs in order to power reaction in our cells using cellular respiration to break up those glucose molecule into ATP. And in that process, we need O2, oxygen, that is provided by the plants. In the end, even though carbon dioxide and CO2 are what's called byproducts for photosynthesis and cellular respiration in the 2 different types of organisms, we actually are dependent on each other to survive because we need those byproducts or ingredients from each other. We need oxygen from plants, while plants need carbon dioxide from us. Okay, so that's the energy cycle there. So this is a good summary chart that compares and contrasts between cellular respiration and photosynthesis. We'll be sure to study this whole chart right here for lecture exam, because this gives you a good view of what's different between the 2 processes, whether it's catabolic, anabolic, what raw materials it starts with, what end products you're ending with, where does it happen, etc. Okay? Make sure you go through this table for your understanding. So this concludes Chapter 8 on photosynthesis, and I will post the answers to practice questions on eCampus.